There are different issues in genome assemblies. Some of them they are related with the graphs. So let's talk about them first. One is spurs, we can have bubbles, we can have converging and diverging paths, and sometimes we observe some cycles. Let's take them one by one. Spurs are short dead end branches, divergence of the main path. So you start from somewhere and then you are diverged. What are the possible causes? There might be sequencing errors towards one end of the read, and maybe it's because of the low coverage. Bubbles are formed if we have a divergence of the path and then afterwards there is convergence. So that kind of arrangement is called as bubbles. Again, it's related with the sequencing errors which are there in the middle of the read now. And there might be polymorphisms in the target. So that can be a reason. So that bubbles are observed in the assembly. Converging and diverging paths. So inverse definition then for bubbles, two paths converge into one and then later on they again split. So that may be because of the repeats in the target genome. Cycles are observed if the paths that converge on themselves. So paths they start converging on themselves, that may be also arise because of the repeats. So how can we control the issues like these we discussed earlier, like bubbles, spurs, cycles, convergences or divergences? One thing what we can do is if we can afford, we can filter the graph of these erroneous occurrences. We can simply discard them or throw them away. In addition to that, there are some other limitations. One of them is loss of information due to splitting into k-mers. So when you are using those deep brown graph assemblies, what we do here is you take those reads and you break them into k-mers. So reads are already short in size and then your k-mers are further shorter. So in this way you might lose some information. Then as far as the repeats are there in the genome, they increase the number of Eulerian paths exponentially. So more repeats, more Eulerian paths, and for that you might have an assembler that is efficient in finding those most probable paths in a real quick time. So for that you might want to increase the memory consumption and the runtime. So obviously these are the issues which pose challenges to your assembly process. So here in this slide we see that sometimes using a shorter k-mer is advantageous over using a longer k-mer. So here for instance we see a read and there is a sequencing error where we see a red color N, so that has been incorporated, which is wrong. So if we use a K3, we will have the k-mers, the small k-mers which are of the length 3. So mostly we can have the k-mers which are unaffected. And if we use the k is equal to 10, so this N will go there obviously, but this is a wrong k-mer. So as far as the true assembly is concerned, because of this one nucleotide is a part of that k-mer, so it will mess up with our assembly. So if our k-mers are smaller, these kind of sequencing errors, their effect can be minimized. So in this section, what we have seen here is that selecting appropriate k-mers is an important steps, and repeats they might collapse the assemblies. We also see that there are some issues which are relevant to the formation of different bubbles or cycles in our graphs. We might trim them.